All right. Good afternoon, everyone, or hello for whenever you're watching this. This is our second to last lecture, and this lecture is going to be covering one of my favorite topics in a first semester biochemistry class, and that is lipids. We're going to talk about a couple of different types of lipids, different classes of lipids, um, and we're going to spend, not a disproportionate amount of time, but most of our time talking about kind of our foundational lipid, and that is the fatty acid. Um, and that's just because it's kind of the most, well, yeah, it's a foundational molecule. So, with all that to say, I'd like to go ahead and get cracking on all this. Now, the first thing that's important for you to know about lipids, and I've broken this chapter into lipids and membranes, so we're going to have a lecture on lipids and then membranes will follow. Now, in contrast to molecule studies in prior chapters, lipids are primarily hydrophobic. So that's an important thing to recognize. Everything that we've talked about, amino acids, for example. There are some hydrophobic amino acids, but then there's also hydrophilic amino acids, your polar positive or polar negatively charged residues, or your polar residues in general like to be in an aqueous environment. Lipids, by contrast, they're very hydrophobic, and part of them would be most soluble in an organic environment. Now, with all of that said, these are different from everything else because they do not form polymers, but do form large aggregates. So they kind of cluster together to form something larger, but it's not a long chain of something. Now, lipids are a very diverse category of molecules related only by their hydrophobic properties, from fatty acids to a variety of hormones. So, yeah, those are learning objectives that we're going to tackle. We're going to get through everything, including protein transporters and different types of transporters. So this is our big hierarchy of lipids. And, as I said, one of the biggest things that we'd be discussing would be fatty acids, because fatty acids are kind of foundational. Now, I would say, or if you look at this model, we've got fatty acids basically on the same tier as steroids, lipid vitamins, and terpenes. Now, these three terms, steroids, lipid vitamins, those might be something familiar to you. For instance, um, if you have ever taken as like a nutritional supplement, or maybe your physician or your PCP has suggested that you take um, vitamin D. Vitamin D is a lipid vitamin. Now, if you've ever seen a tomato or something like that, tomato has a robust red pigment and carrots have an orange pigment because of terpenes. Steroids, well, typically when people think of steroids or talk about steroids outside of a academic setting, like, generally that's going to be something about performance enhancing drugs or something like that. Now, where we're going to get started is not in that block, but instead we're going to get started with this discussion, fatty acids. Now, fatty acids, think about that as a big umbrella. Underneath that umbrella, you've got eicosanoids, glycerophospholipids, triacylglycerols, waxes, sphingolipids. So those five different major topics, well, they have some things underneath them, but they all fall under the category or the umbrella of fatty acids. So without any further ado, let's figure out what a fatty acid is. A fatty acid is a molecule with a non-polar tail, a hydrophobic tail. And if you look at this molecule above, and you look at this chain right here, I'm sure that you are all knowledgeable enough to see, well, that chain right there is just a bunch of carbons and hydrogens. You said that, you're not wrong at all. Now, the other part of our fatty acid is right here. It's a polar head group. So that's going to be something that's actually soluble in an aqueous environment. It's water soluble. So we have a molecule here that's got one end that's a long hydrocarbon tail. And then we've got another end that's a polar head group. So we've got this molecule that's made up of two major parts. One part is very hydrophobic, the other is very hydrophilic. Okay, so something is water hating, and then the other part is water loving. So these are important traits of a fatty acid, or important properties to understand about fatty acid. Now, commonly, so your most common fatty acid, these tails are usually straight chains. Most range from 10 to 20 carbons. Usually, they have an even number of carbons, including the C from the carbonyl group. So, even number, we're talking about 16, 18, 20, 22. Okay, something other than that. An odd number like 3 or 15 or 17. We typically don't see those very often. Now, is that good? Is that bad? Is that anything? It's not good. It's not bad. It's just a simple fact of the matter. Odd chain fatty acids are not as common as even chain fatty acids. Now, next up is when we talk about this long fatty acid chain, these carbons and hydrogens. Okay, so we fill in, there we go, there we go, there we go. These chains can be saturated, unsaturated, or polyunsaturated. So when we talk about those different terms, saturated, unsaturated, and polyunsaturated, let's look at these fatty acids right here. Here, what we have are a total of four different 18 carbon long fatty acids. One has the common name of stearic acid. The next is oleic acid, linoleic acid, and alpha linolenic acid. These are four different fatty acids. What I want to draw your attention to is the top of them. They all have this carboxylic acid right here. So that part of the molecule effectively is water loving. It's soluble at a physiological pH in an aqueous environment. Now, what I want you to notice about all those is that is carbon number one. Now, if we look all the way down at the bottom, we have a methyl group. That methyl group is labeled 18, not because we're dealing with 18 isotope of carbon, but instead, this carbon is number 18. So these are all fatty acids that are 18 carbons long. Notice how they get progressively shorter, which is interesting because it's kind of realistic. Um, but what I also want you to notice about them getting progressively shorter is that there's a reason why. And that reason is we're introducing one double bond, two double bonds, or one, three double bonds to constitute that or those differences. Now, if we can think about that, and then we bounce back to our terms here, saturated, unsaturated, or polyunsaturated. Now, I'm going to tell you that when you look at these four different structures, there's one that is saturated, one that is unsaturated, one that's saturated, one that's unsaturated, and then there are two that are polyunsaturated. Okay. Now, you'd be correct in saying that linoleic and alpha linolenic acid are two polyunsaturated fatty acids. These are fatty acids that have more than one double bond. Now, what's important to notice is that whenever I say one double bond, I'm of course referencing in the hydrocarbon chain. I don't count that carboxylic acid as having a double bond because of the fact that, well, chances are that group is going to be resonance stabilized. So it really doesn't have a double bond. Instead, it's got kind of like a sort of double, sort of single bond. Okay. So when you talk about a polyunsaturated fatty acid, you're talking about a molecule with two or more double bonds in that long hydrocarbon chain. Now, the next thing that's worth noting about these is that double bonds are usually cis. There are trans double bonds, and that sometimes is produced as the result of a cooking process. If you go to McDonald's and many other restaurants or many other food vendors, they'll say something like, "No trans fats produced in the production of this food." Um, and that's important because trans fats are metabolized much differently than cis fats. So trans fats don't really your body doesn't process them the same way. And they can present health risks or health problems. So these double bonds are not conjugated also. So one of the things that's worth noting is that if you look at linolenic acid and alpha linolenic acid, what you notice about linoleic acid is that we have a pattern of a double bond followed by two single bonds, and then there's your double bond. Now, if you look at alpha linolenic acid, you'll see that same pattern: double, single, single, double, single, single, double. That is an indication that you have a fatty acid that is not conjugated. That's what that term non-conjugated or unconjugated, what that means. Okay. Now, when we think about these fatty acids, there's some unique physical properties about them. For instance, their melting points. They all have the same number of carbons, but they have different numbers of double bonds. The different numbers of double bonds are going to influence their melting points. So those melting points are going to go down as the number of double bonds is present. Okay, so humans can synthesize all except two fatty acids: linoleic acid, an omega-6 fatty acid, and linolenic acid, an omega-3 fatty acid. Linoleic acid or linolenic acid is an essential fatty acid. It's found in plant and fish oils, and it's necessary for regulating blood pressure, clotting, lipid levels, immune response, and inflammation response. Now, here what we have is another rundown of all these different fatty acids. What I'd like to draw your attention to are basically three different rows. I'm having trouble finding. There we are. There's my cursor. The first one that I'd like to draw your attention to is right here. 16 number of carbons, 16. Number of double bonds, 0. Common name is palmitate. This is sometimes, or you will sometimes hear it referred to as palmitic acid, but palmitate is your ionic form of this fatty acid. The melting point of palmitate is 63 degrees Celsius. What that means is that we're just to kind of put it into perspective, at room temperature, palmitate will indeed be a solid. Okay? Now, so palmitate will be a solid at room temperature at 23 degrees Celsius because it's going to stay a solid until it hits 63 or more degrees Celsius. Now let's jump down here. 16 carbons in that chain and then one double bond. That fatty acid is known as palmitoleate. Oh boy, that's a complicated name. Palmitoleate. So the difference between palmitate and palmitoleate is a single double bond. Palmitoleate has a melting point of negative 0.5 degrees Celsius. Okay, so when we introduced that one double bond, our melting point went from 63 down to negative 5. So essentially what that means is that if you had palmitoleate at room temperature, it would 100% be a liquid. Now, if we move up one row, we've got an 18 carbon long fatty acid with a single double bond. Oleate is the name of that. Oleate has a melting point of 13 degrees Celsius. So if we compare oleate to stearate, which has a melting point of 70 degrees, we see again the introduction of a single double bond causes that melting point to just plummet. Now, a great question for that, oopsies, is why? Well, one of the reasons is when you look at a saturated fatty acid, so one with nothing but double bonds, you can pack those really well together. They form a more cohesive solid. Whereas all of these fatty acids with at least one double bond, they're going to kind of push off from one another. So they're all going to be kind of assertive and say, like, well, I need space. And if you have a bunch of those, well, then you're not going to form something that is a solid indefinitely. So these polyunsaturated and monounsaturated fatty acids, they occupy a lot more space, so they can kind of like push one another away. So they don't have those strong cohesive forces through the intermolecular forces that um, your saturated fatty acids have. Okay. The next molecule that we're going to talk about, which arguably is like as close as a fatty acid is going to get to being a um, as close as a fatty acid is going to get to being a part of a polymer, or a lipid is going to get to being a part of a polymer. That's a triacylglycerol. A triacylglycerol takes a molecule like this, glycerol, and sees those hydroxide groups, and for each one of those hydroxide groups throws a fatty acid on there. And we end up with something that looks a little bit like this. On these different carbons, you're going to have these ester linkages in the long fatty acid, long hydrocarbon chains. So this is a long hydrocarbon chain. This is a long hydrocarbon chain. This is a long hydrocarbon chain. So these make up a triacylglycerol. Now, there are types of membrane lipids, or major types of membrane lipids. You've got cholesterol. Cholesterol, if you look at that, man, that doesn't look like a fatty acid at all. Is that wrong? Nope, because cholesterol is a steroid. So cholesterol finds itself over on the right here as a part of the isoprenoids. Isoprenoids are those molecules that make up vitamin D3, make up pigments of carrots and tomatoes, and yeah, and they also make up, yeah, cholesterol. Now, phospholipids are an interesting class of molecule because what they have is they have two groups that are just your regular run-of-the-mill fatty acids. You have glycerol as a backbone, and on that glycerol, you're going to have a phosphate alcohol. Ooh, so it's going to have a phosphate and then a hydroxide group. Okay, so that's our alcohol. A glycolipid is going to be something that is basically a fatty acid unit that has been added to a cerebroside. That cerebroside has a sugar, like glucose or galactose, linked to that. Now, those get much more complicated, and we'll take a look at those in just a little bit. But in the meantime, these are our phosphoglycerides, also known as our phospholipids. Now, I said that we've got a um, we've got a glycerol backbone, we've got our two fatty acids, we've got a phosphate, and then X, just the filler. Well, that X could be any one of these molecules, making any one of these particular structures and these particular molecules. So these are embedded as a part of a membrane. Now, what's pretty cool about these is that all of these different glycerophospholipids right here, they do a number of different things. Some of them are going to be on one side of a membrane, but then when a cell is uh, pressured or attacked, well, then what's going to happen is that phospholipid is going to end up on the other side of the membrane and be able to communicate with the outside parts of that cell. Sphingomyelins. These are molecules that contain a sphingosine backbone. Sphingosine is 18 carbons long, and when you look closely, it kind of resembles the backbone for a glycolipid. So, where are we? Sphingosine has this lengthy carbon that's got a hydroxyl group, carbon number 1, where this NH3 is. Lo and behold, that's a good place to put another fatty acid chain. We've got our phosphate group where we add an amino acid to that phosphate to make a molecule like phosphatidylcholine. Um, okay. Now, glycolipids are carbohydrate-containing lipids derived from sphingosine. Okay. The carbohydrate, so here's our sphingosine. We modify this to make a glycolipid. The carbohydrate is linked to the primary alcohol of that sphingosine. So that primary alcohol was on carbon number 1. So that's where we're going to add in a sugar like galactose. Now, cerebrosides are the simplest glycolipids because they contain only one single sugar there. So it's going to have, so like what we see here, this is an example of a cerebroside. Now, gangliosides are basically this, but more complicated. Gangliosides contain a branched chain of as many as 7 different sugar molecules. Now, the powerful thing about these is that this end of our molecule, I'm going to highlight my two ends. So those two ends right there, those are very hydrophobic. Those ends can insert into a lipid bilayer. Now, that's all good and well, but what that means is that anything where that primary alcohol is, in this case, that beta-galactosidase has been added, well, that's going to serve ultimately as a mechanism to communicate from one cell to a neighboring cell. Now, what I have circled right here, 3, 2, 1, just counting down so everyone's had a chance to look at it. What I have highlighted right here is, I gotta find my cursor, is this right here. In the case of a ganglioside, we have that primary alcohol, which has been replaced by glucose, and a chain that goes on a little bit. So glycosphingolipids on cell surfaces act as receptors and they act for recognition. So these gangliosides are tools that cells use, again, to communicate. In addition to that, these gangliosides are processed as a part of neurological development. And what happens is that these sugars get clipped off in different sequences as a means of saying, hey, this cell's ready to divide, or this cell's ready for this, or it's ready for that. So ganglioside degradation is a very important part of um, ganglioside degradation is a very important part of neurological development. And in the event that part of ganglioside degradation is disrupted, well, then there are a number of different diseases that develop as a part of this. Ganglioside development is also tightly associated with lots of people's understandings of, or the general understanding of genetic linkages to diseases, because there's lots of genetic disorders that are related to ganglioside degradation because it's an issue with an enzyme or issue with enzymes that lead to lack of ganglioside degradation. And as it turns out, it's, you know, anyone who has an ancestor from Eastern Austria, for instance, they could have this disease, and it's because of a genetic mutation. Okay. Steroids are molecules that have three six carbon structures, three six membered rings, and one five membered ring, as is indicated here with this ABCD ring structure. Now, cholesterol is the precursor of many steroid hormones. So when you look at this four ring structure, there's a number of different modifications take place at these different positions on these different rings. So carbon number 8, for instance, well, if that has a methyl group, that could be a performance enhancing drug. Depends on what it's going to lead to the production of, or what it's going to lead to with respect to, gene regulation and expression. Now, here are our major steroid hormones, and their progenitor molecule, or kind of where they started out from. That's right here, squalene. Squalene is a compound that is involved in cholesterol biosynthesis. See, if you kind of squint your eyes, it's almost like you can see the rings beginning to form. So these rings in squalene, um, or squalene is a molecule that precedes cholesterol in cholesterol biosynthesis. And that's where I'm saying if you kind of squint your eyes, oh yeah, there's the different rings. Um, Cholesterol depicted here in the upper right hand corner is the molecule that is metabolized for the production of things like testosterone or sodium cholate or ergosterol. These are all fatty acids, or sorry, not fatty acids. These are all lipids that are produced for different purposes. Testosterone is an example of a steroid hormone, and it's naturally produced by people's bodies, and it's set in balance with other things, with other steroid hormones. But some people take nutritional or not nutritional supplements, they'll take injectable testosterone so that they can, lift more weights or something of that nature. Okay, so there's a couple of other lipids. We've got our waxes, eicosanoids, and isoprenoids. If you look at isoprenoids at the bottom here, isoprenoids are a class of compounds that basically have these long chains built from a molecule that looks like this one right here. It's 5 carbons long, and basically you can take this and build it. And I always kind of think about this as being almost like a Lego. You stack one, and you stack another, another, and you can make a long chain. When you make that long chain, what happens is you get a molecule like squalene. That squalene can then morph into something like cholesterol. So this complex structure of 20 some or 27 some odd carbons that make up that four ring structure of cholesterol, that's really coming from a bunch of additions of that molecule that I've got highlighted at the bottom. Waxes are another class of compounds. Waxes are found on birds' feathers, and they help water just kind of bead off of them. Eicosanoids, well, here's an example of an eicosanoid. One example of an eicosanoid is the fatty acid known as arachidonic acid. I think that's maybe one of the coolest fatty acid names that I can think of. Um, in part because it sounds like it's just like a band name or something like that. So, arachidonic acid is a fatty acid that gets cleaved and is acted on by an enzyme known as PGH2 synthase. That PGH2 synthase is then acted upon to produce prostacyclin, prostaglandin, thromboxane. All three of those are produced as soon as this PGH2 is produced. Well, if you look at PGH2, what you see is that this 20 carbon long molecule has basically adopted a ring. And the enzyme that does this, does that ring formation, is in the class of cyclooxygenases. So if I remember correctly, PGH2 synthase is a cyclooxygenase. Now, those ring structures that are going to form are going to lead to the production of these metabolites shown at the bottom: prostaglandin, prostacyclin, thromboxane. Well, those are all compounds that uh, lead to pain and fever and blood pressure increase and blood coagulation. So we, as a population, typically will use something like, in the event that we have like pain or something coming on, we'll take aspirin. That aspirin will block the cyclization of arachidonic acid into the cyclooxygenase. So that aspirin blocks a cyclooxygenase. So it's a cyclooxygenase inhibitor. Okay, that's going to wrap up our conversation, or what we need to know about um, fatty acids and basically the first part of lipids. I will put together your conversation or your lecture on membranes and what make those a little bit different in the coming day or so. Um, but make sure that you know how to classify fatty acids and think about different melting points. Physical properties are important. We'll get into that a little bit more when we're talking about membranes, but for the time being, I hope you have a good one. All right.